Welcome to the deep dive. Today we're tackling something really counterintuitive about our immune system, specifically T cells.
Right.
When they get exhausted fighting cancer or chronic infections, you'd think they'd, shut down manufacturing to save energy.
That's the logical assumption, the standard stress response.
But this paper finds the complete opposite happens. These tired T cells massively ramp up protein production, and it actually ends up being self-sabotage.
It's a critical finding because it really forces us to rethink what T-cell exhaustion even means at a molecular level.
Wow.
We know these CD8 plus T cells, the effector T cells, or TEFs.
The fighters?
Exactly. They shift into this dysfunctional state, we call it Tex, in chronic diseases. And that's a huge roadblock for immunotherapies, things like checkpoint inhibitors.
Yeah, therapies that try to wake these cells up. And okay, traditionally people have focused on surface markers like PD-1, right, and maybe epigenetic changes.
That's been the main story. Inhibitory receptors, constant antigen exposure, changes to the DNA packaging. But the role of protein quality control was scientists call proteostasis. That was largely missing.
The cell's internal factory floor and cleanup crew.
Precisely. And the authors pointed out something important. Looking just at RNA messages wasn't enough. They found a surprisingly weak link, like only R0.3 to 0.4 correlation between the RNA instructions and the actual protein levels in these T cells.
So the blueprint doesn't fully predict what gets built? Okay, so they use a technique called quantitative proteomics, mass spectrometry.
Yep, to directly measure the proteins. They compared exhausted Tex cells from mice with chronic viral infections and also some tumor models against functional Tef cells.
And what did the actual protein landscape reveal?
It was striking. Tex cells showed this really marked specific increase in proteins involved in the proteotoxic stress response, or PSR. They actually coined a term for it. Tex-PSR.
Tex-PSR. Okay.
This meant the cells were churning out specialized helpers, chaperones, located in the endoplasmic reticulum, things like BP and GP96. Their job is to help fold proteins correctly.
Or tag the bad ones for removal. So the cell definitely knows it's in trouble. It's sending in the quality control experts.
It knows. Which brings us to the core puzzle here, and maybe a thinking question for you listening. If a cell is under this kind of severe stress, it's got misfolded proteins piling up, it's deploying chaperones, should its overall rate of making new proteins be high or low?
Intuitively, I'd say low. Right. Stop the production line, clear the backlog, conserve energy.
That's the intuition.
Yeah.
But when they measured it, first, they confirmed Tex cells did have a significant buildup of protein aggregates, clumps of misfolded effector molecules like granzyme B and perforin, the very weapons the T cell needs.
Oh, wow. So it's making the weapons, but they're faulty, and they're literally clogging up the system, making the cells sick.
You got it. Toxic internally. And here's the kicker. Despite this mess, Tex cells showed a significantly higher rate of global protein synthesis compared to the healthy Tef cells.
No way. So the factory's running faster than ever, even though it's producing faulty goods and the quality control's overwhelmed.
Exactly. It's this bizarre paradox. The system is stressed, the chaperones are active, but protein production is actually accelerated, making the problem worse.
Okay, that leads straight to the big question. Is this Tex-PSR thing just a symptom of exhaustion? Or could it actually be causing it? How did they figure out which came first?
They tested causality directly. They took healthy Tef cells and basically forced them to experience protein misfolding, either using chemicals or by making them express a protein known to aggregate, like a mutant CFTR protein.
So they deliberately gummed up the works internally.
Right. And just doing that, inducing proteotoxic stress alone, was enough to make the healthy T cells become exhausted. They started showing the hallmarks, like expressing PD-1.
That proves it's causal then. It's not just a side effect.
It seems to be a driver. And they even traced it upstream. They found that sustained activation of a signaling pathway called AKT, that was the key trigger, forcing AKT to be constantly active, drove this whole Tex-PSR program, the high translation rate, the aggregation, and the full exhausted state.
Okay, let's zoom out. This feels like a pretty fundamental shift. This work is saying proteotoxic stress isn't just like a footnote. It's a whole separate mechanism, maybe a third pillar alongside the known gene control and epigenetic stuff that keeps T cells exhausted.
Exactly. It reframes exhaustion. It's not just that the T cell is tired or suppressed. It's almost like it's addicted to this dysfunctional high-speed protein production system that's ultimately harmful.
And the implications. Are there therapeutic angles here?
Absolutely. This is where it's really exciting. They found that if you specifically targeted or inhibited those key chaperones, GP96 and BP, in preclinical models.
The ones that were upregulated in the Tex-PSR.
Yes. Inhibiting them actually reversed T-cell exhaustion. It improved their ability to fight tumors. And importantly, they also saw this effect in human T-cells. They developed a Tex-PSR signature score.
Like a measure of how active this pathway is.
Right. And in T-cells taken from human cancer patients, a high Tex-PSR score strongly predicted that those patients would respond poorly to current immunotherapies like anti-PD-1 or CAR-T cells.
That's a huge clinical connection. Okay, so wrapping up the key takeaways for everyone listening. First, T-cell exhaustion involves this unique proteotoxic stress response, the Tex-PSR, marked by paradoxically high protein production and misfolding.
Second, this proteotoxic stress isn't just correlated, it's causal. Forcing protein misfolding is actually enough to induce exhaustion in healthy T cells.
And third, this whole pathway is potentially druggable. Targeting specific chaperones like GP96 or BiP might be a new way to restore T-cell function and maybe boost existing immunotherapies.
It opens up new therapeutic avenues. But there's a final thought, a tricky balance, isn't there?
What's that?
Well, if just stimulating T cells more, maybe through pathways like AKT, actually worsens exhaustion by fueling this proteotoxic fire.
Right, you make them run faster into the wall.
Then the big challenge becomes, how do we fine-tune this? How can we manipulate AKT or the PSR machinery to make T cells effective killers, but also durable without causing unintended side effects or toxicity? That's the next frontier.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.